Hi everyone and welcome to the internet. What we're going to be doing in today's lesson is having a look at decomposers. So hopefully by the end of this lesson you're going to be able to describe what decomposers are and explain how the process of decomposition actually takes place. To start us off though, we've got 3 questions based around our previous lesson, so have a go answering these 3 questions in your books. Now for question 1, do try to give as much detail as you can, definitely including the names of those processes. So I'm going to give you about 5 minutes to have a go at this today. Okay, let's go through those answers then. So obviously any of the points that you've got, make sure you give yourself a tick so that you can get a mark at the end of it. So the first one, to explain the carbon cycle, then there's a lot of marks here. So if you've said that carbon dioxide in the atmosphere is taken in by plants, give yourself a tick. If you said that the process that does that is photosynthesis, tick for that bit. And if you said that that carbon dioxide is used to make glucose, give yourself a tick for that. Now you may have done that as the word or the balance symbol equation for photosynthesis, absolutely fine, you can get that mark there. You then go on to say the plants are eaten by animals, tick. So the carbon compounds move along the food chain, tick. However, if you said carbon dioxide moves along the food chain or carbon dioxide moves from the plants to the animals, you don't get the mark, because carbon dioxide is converted into compounds like glucose, cellulose and so on in the plants, and those carbon compounds are what transfer to our animals. So just go careful to phrase it that carbon compounds move from the plants to the animals. Those plants and animals carry out the process of respiration, tick, releasing carbon dioxide into the atmosphere, tick. You can also talk about the fact that plants and animals die and then become decomposed, tick for that bit, which releases carbon dioxide from those decomposers carrying out respiration of their own, tick for that. You then get a mark if you said that fossil fuels are burnt or combustion of fossil fuels, and then your final mark there is for saying it releases carbon dioxide back into the atmosphere. For question 2, the reason that we've seen the fluctuation or the increase in our carbon dioxide over the past 100 years is we've seen an increase in cars or vehicles or transport, that's fine, and factories. So those two get you a mark, one each, and this is for linking it into the fact that they release carbon dioxide through the burning of fossil fuels. The last one then, question 3, is why we get that variation across a 24-hour period. Your first mark is for pointing out that photosynthesis only happens during the day. Second mark is for saying that carbon dioxide is only removed in the day. Third mark is that respiration happens all the time. And the fourth mark there is so carbon dioxide is always released. So add up your total score for me and write that in your books. We've got a total of 17 today. So give yourself a score out of 17 in your books. The main thing we need to understand today is the difference between two different types of organism. The first one are decomposers, and the second, the detritivores. So we're going to kick off with the decomposers first of all. Now what we've got on the screen here are 4 sentences that are only part finished. All the words you need to finish them off are in the red box at the bottom. So what I'd like you to do is write those sentences in your books and have a go at finishing it off with the words in the word box at the bottom. So I'm going to give you about 4 minutes to have a go at that. Let's check those answers then to make sure we've got it right. As always, if you do have it right, give yourself a tick. If you didn't get it right, make sure you put the correct answer in so you've got a complete set of notes. So decomposers are microorganisms. They are bacteria and fungi. You could have had that as fungi and bacteria. Either way around works there. They break down or decay dead organic material as well as feces and urine. Again, feces and urine can go the other way around to be urine and feces. And this returns nutrients to the soil. So what we see here, when we're looking at the decomposers, these are microorganisms, so things we can see using a microscope. Now, the two we need to remember are bacteria and fungi. And the way that they actually work is by breaking down this dead organic material and the feces and the urine. And through that process of decomposition, we're returning those nutrients that were trapped inside that dead animal or the feces and the urine it left behind into nutrients that then go into the soil. So make sure you've got those written down. I'll leave it up for another 10 seconds just in case you need any more. We do have another key word that we need to know related to our decomposers, which is the word saprophyte. Now, saprophytes are just a type of organism, and they're a type of organism that feed on this dead material or waste, and the way they do this is that they actually secrete or release the enzymes from themselves onto the surface of that dead animal. Now, once the enzymes are on the surface of the dead animal, what happens is that they're going to break it down, and then the saprophyte absorbs the nutrients. So what we're seeing here is, it's not a process of digestion like we find in humans, where it takes place inside a body. Here, our saprophytes secrete the enzyme onto the surface of the dead thing, the enzymes then break down those dead remains, and then they take up those soluble nutrients afterwards. So make sure you've got those two sentences written down. I'll leave them up for another minute for you. So we've looked at our decomposers first of all. Now we need to have a look and see what a detritivore is. And don't mix these up, okay? They're two very different organisms. So decomposers, the bacteria and the fungi, detritivores are small animals. And the way that they help decomposition is very different. So what the detritivores actually do is they break up that organic material into really small pieces. Now, by breaking up the organic material into very small pieces, then what they've done is created a much larger surface area. When we've got a larger surface area, there's obviously more space for the decomposers to work on, and therefore decomposition is going to happen faster. So what we see here is the detritivores and the decomposers are actually kind of linked in the way that they work. So the detritivores are small animals that are going to break up those large chunks of organic material into these small pieces, and then the decomposers will work on that large surface area of those organic materials and release the enzymes onto the surface. The enzymes break down the dead material, and then the soluble nutrients are absorbed by the decomposer. So the two things work together, but they are not the same and they work in a different way. So make sure you've got these 3 sentences about our detritivores written in your books. I'll leave them up for another couple of minutes so you've got a chance to really get them in your books there. You do need to know the examples of detritivores as well. And so here are 3 really common ones that you would find in any habitat in the UK particularly. So first of all, if you go out in your garden and dig a hole somewhere, you're bound to find earthworms. Now, earthworms are detritivores. They're feeding on dead organic material. And the stuff they're normally feeding on are leaves that have fallen out of the trees onto the ground. So what they do is they actually take in those large bits of leaf and then they pass out their other end of their single digestive system, these small pieces which have got the large surface area. We've got woodlice that are going to obviously work on wood. That's why if you turn over an old log or a bit of bark outside, then you're going to find woodlice under there because they're actually feeding on that. And then we've got maggots, which you'll find on any dead animal lying around. So hopefully you don't have too many of those lying around in your garden anyway. So make sure you've got those 3 examples written down for me: earthworms, woodlice and maggots, and what they're actually going to be breaking down. The next bit I've got for you is something that if you don't like slightly gruesome things, then you may not want to watch it, in all honesty. We're going to actually have a little look at a time-lapse video of what happens to a dead animal. Now, we've got a monitor lizard in our video here, so what we're going to do is watch what happens to the monitor lizard over a period of time. So I'll give you 1 more minute to write these down and then we'll watch that video. Obviously, if you don't want to, then if you skip forward about a minute at the end of this section, you won't have to watch it and you'll be on the next slide. Okay, so now we've seen that lovely video of a decomposing fox, then if you particularly want to watch more decomposing creatures, then by all means feel free to search on YouTube for decomposing organisms. There's many things you can see. Now, what I'd like you to do then is just thinking about our decomposition. There are certain factors which affect the rate of decomposition. So 2 questions on the screen, have a go at answering those in your books for me. I'm going to give you about 3 minutes to do that. Okay, let's check your answers then. Hopefully the factors that we've got that affect the rate of decomposition are temperature, the moisture content, and the amount of oxygen. Now, in terms of the explaining part, then what we need are warm temperatures first of all. That's the best, because if we actually have the temperature too high, then what we find is the enzymes within our decomposers will become denatured. And if we think back to our work we did in our early biology topic on enzyme reactions and temperature, then denaturing means the shape of the active site no longer fits and the substrate, and therefore it doesn't work. So denaturing, bad. Too high temperatures, bad. If, however, the temperature is too low, then what we see is that the rate of those enzyme-controlled reactions are very slow, they're reduced, and that means that decomposition is going to take a really long time. So that's why warm temperatures are best. Not too hot, not too cold, kind of a Goldilocks experience. In terms of the water content, then our microorganisms actually need water to carry out the decomposition. So what we find is that if our microorganisms don't have water available to them, then reactions within them won't be happening or will be happening at a very reduced rate, and that means that decomposition won't take place. So again, best is a moist environment. And then in terms of the oxygen, for decomposition to happen well, we need plenty of oxygen available, because oxygen is used in aerobic respiration. And again, go back to our first biology topic. Hopefully we remember that glucose plus oxygen makes carbon dioxide plus water, and it generates the ATP they need for all of the other reactions. So if we don't have enough oxygen, then it won't be able to carry out aerobic respiration, which means it's not going to survive. What we find is that if we've got anaerobic conditions, i.e. there's not enough oxygen available, then decomposition generally doesn't happen, which is what we see in things like waterlogged soils where there's just too much water and there's not enough oxygen there. Again, decomposition can't take place. So the best conditions we can have for decomposition: warm, moist, and plenty of oxygen. And watch out for these. They do like questions asking about the conditions for a compost heap and how best to decay the material within it. So just make sure you do know those 3 factors there. If you've not already got it written down, I'm going to leave it up for another minute for you. But as always, if you need more time, just hit pause on the video. The last bit we're going to look at today is how we calculate the rate of decay. And what I've given you on the screen then is the formula we need to do this. So the rate of decay, which we usually use the units in grams per day, is the change in mass, obviously in grams, divided by the time in days. So write that formula in your book so you've got it there to refer to. So you've got the formula. Now have a go at answering the question on the screen. So I'm going to give you a couple of minutes to have a go at this, then I'll show you how we do it. So let's go through how we actually work out the answer to this question then. Now in the question it tells us to compare the rate of decay of bread between the first 5 days and the last 5 days. So what we're really looking at are two sections, so the first 5 and the last 5. So what we need to do first of all then is remember our actual formula that we're going to use. So our rate is the change in mass divided by our time. So that's what we're going to use throughout this. Now what we need to do is calculate our two rates. So we need to calculate a rate for the first 5 days first of all. So what we can do is for the first 5 days is we're going to be looking at the change in mass. So that's between 0 and 5. So what we need to do is 30 minus 28 to give us our change in mass, and then we divide that by our 5 days. So when we do that, that works out 30 minus 28 is 2, divided by 5 gives us 0.4. So our first one is 0.4 grams per day for the first 5 days. Then we do the same thing, but this time for the last 5. So if we have a little look between the last 5, it's here. So it's 20 minus 10 to get our change in mass. Again, divided by 5. Hopefully we know that 20 minus 10 is 10. Divide that by 5 gives us 2 grams per day. Now that's not the end, because the question, remember, was to compare the rate. So we've got our rate for the first 5 days of 0.4 and then our rate for the last 5 of 2. Now hopefully we can see that actually the last 5 is happening at 5 times the rate of the first 5. So all we need to do at the end is say that the last 5 days decays at 5 times the rate of the first 5. And that's it. The key thing to watch out for these ones is don't just say that the last 5 days is faster, because we've actually got numbers. We can work out how much faster it is. So don't just say it's faster, say the last 5 days decays at 5 times the rate. So in terms of how we can get our marks, so I'm going to give you a mark if you got your formula. You can have a mark if you had the working here and a mark for this bit. Then the same, mark for your working, mark for the answer, and then a mark overall there, which gives us a total of 6 marks. So give yourself a score out of 6 for that one for me. If you want to see any other videos of various organisms being broken down by things like our Detritivores, then just search for them on Google and on YouTube. You'll find many different animals being broken down in time-lapse videos. So last thing for us to do today then are our 3 plenary questions. So I'm going to leave these up for about 5 minutes today, but as always, if you do need longer, then just hit pause and take as long as you need. As always, you can find the answers to the plenary over on my website, scienceright.weebly.com. You'll also be able to find that under the Internet tab. Just select the week that we're in, and then you'll be able to find things like the lesson PowerPoints, the answers to the plenary, any additional work, and if there's any little experiments I thought you could do at home, then they're there too. So have a look and get using that.